go ahead and get started on a couple of things on As You Learn. Okay, so the first thing I want to do is I want to talk through the material with you guys. You may have or may not have watched Parks' video. If you have, that's great. If you haven't, this is for everyone else who hasn't yet. And we're just going to kind of talk through it. So let's do that first. Okay. Now, can you guys see my screen pretty well right now?
If we're looking at white background,
It's still loading, but you should be able to see
You're lagging pretty bad,
it.
Srudasa.
Screen share should be up. Yeah. Yeah, it's taking a moment to load. We're still having a couple internet connectivity issues this way.
You're lagging.
Well, man, what are you doing? Chill out. Okay? Chill for a second. Come here. Calm down. Calm down. Come on. Yeah, it's still loading. Come on. Now, this gene is heavily reliant on altering things within the circadian clock. It's endogenous within us timekeepers that control our physiology and behavior. That means it works on a 24-hour period. It's synchronized by external cues, especially sunlight, and there may be involuntary rhythmic outputs or changes as an effect of change.
Excuse me?
Mhm.
We haven't been able to hear you for the past few minutes, and we can't see your screen.
Yeah, I can hear you fine, but I can't see your screen.
That's
I
lovely.
just started hearing you.
Same, twin.
Yeah.
Well, I'm glad you said something. So we'll just start this last slide over again. Give me 1 second. Okay.
I can see your screen now.
Yeah, I can
Yeah,
too.
same.
All right, good. Sometimes when the internet kicks off for a second or it starts back, sometimes the screen will break. So hopefully that's a little bit helpful. But basically, the premise of this question is, are you a night owl or a morning lark? Okay, it's a motivation for a lab exercise where a correlation between genotypes and sleep phenotypes as our observed trait or behavior need to be evaluated. So this gene, called the PER3 gene, is heavily influenced by something called a circadian clock. This is an endogenous within a timekeeper controlling physiology and behavior that works on a 24-hour time period. It is synchronized by external cues, especially sunlight, and there are many involuntary rhythmic outputs as an effect of changes to the circadian clock. Okay? The biggest thing that we are looking at here is called gene expression. So, now for class, there are a couple of guiding questions. Is there a relationship between a person's or genotype and their phenotype or their tendency towards morningness? Can we extract this DNA and amplify a gene to show that person has a preference for morning or night? So, in eukaryotic organisms, DNA is stored within the nucleus of the cell. This is a molecule that carries genetic information for each individual organism or stores biological information. Okay? It's made up of macromolecules, so deoxyribonucleic acid monomers, okay? And it has 3 parts. Those phosphate groups, okay, that make it negatively charged, and you can see that here with oxygen. And this also makes it water soluble. Our deoxyribose sugar backbone and our nitrogenous base, okay? And this allows hydrogen bonding to occur between monomers, okay? So, a single strand of DNA looks like this, but it is made up of 2 complementary strands. When we say complementary, we mean that A attaches to T and C attaches to G. So you can kind of see that bonding happening here. And A, T, C, and G represent different monomers or bases that make up the sugar: adenine, guanine, cytosine, and thymine. When these strands are split apart, you can see what their sister strand or sister attachments would be based off of the orientation of the molecule. So, genes are a basic hereditary unit which are made up of DNA and act as instructions to make proteins. They are located on chromosomes. Humans have 23 pairs of chromosomes, with each chromosome containing between 60,000 or 60 to 2,000 genes. So, here you can see an X chromosome, which is a tightly wound piece of DNA within the nucleus. Okay? And you can see this here. So, part of the interesting thing about a chromosome you may not know is the DNA chromosome is wound up into itself by these molecules that look like big yellow blobs, and they kind of come in and they wind themselves all around it, okay? So that's how we get this shape here of this molecule. So, gene expression. Gene expression is a process by which the information that is encoded in the gene is used to assemble a protein. Okay, although not all genes code for proteins, some help with regulating the activity of other genes. So I know I may not have talked to you guys a lot about the research that I'm doing, but actually there is a gene that we do a lot of work with called the FOR gene, or the 4 gene. It's both regulated and both regulates and is regulated by a gene called FOXO. Genes that control other genes or impact the activity of other genes are called controller genes. So yes, there are genes that regulate or control other genes. They tend to be especially involved in development. There are different versions of genes or genotypes that can lead to different appearances or changes in function or phenotype. Okay, you can kind of see this replication process happening here. We have that double-stranded helix of DNA. We have a replication fork that is opened up on both sides into a replication bubble. We have mRNA that is being formed and attached based along one specific point where it has bonded within that fork, and then new bases are built or added in. We end up with a full and complete strand of mRNA, which we then have proteins that come in and can attach at certain spots, allowing for translation of this to occur and for this to be transformed into a protein. So this happens through tRNA and tRNA conversion to protein strands. So yeah, it's a very, very long process, and I'm sure you've talked a lot about in class. And if you haven't yet, this is a basic introduction to it. Make sure when you go over it in class, you go over it into more detail, okay? So, can you guys still see the screen? How is everyone doing?
Yeah, I can see it.
Same.
So the PER3 gene influences circadian rhythms again. The circadian blocks or clocks operate at a cellular level in the brain, okay? The circadian clock is controlled by a genetic feedback loop that we can kind of see happening here, okay? So proteins regulate the expression over a 24-hour time period. And by feedback loop, we basically mean that the products of this loop feed back into the loop and cut the loop off from continuing. In this case, it is a negative feedback. So when these products come back in, this process will stop. If it's a positive feedback loop, then the products will and allow for the process to continue at a greater level at which it was previously. You can kind of see that. So this gene has been found in humans and has been shown to influence an individual's circadian rhythm again. So some genes have different alleles or variations. So how many of you guys know what an allele is? Have like a basic understanding of what an allele is? Okay, so alleles are different versions of the same gene. So we may have a gene for PER3 or anything, right? So let's just look at eye color again for an example. The orientation of the eye, the placement of the cornea, and the color of the eye are all controlled by the same long segments of gene. There are variations in this gene, okay, that can impact, whether or not you are nearsighted or farsighted, but also eye color. So different versions of the same gene that can kind of express themselves in differential means and make different protein products that all have the same basic genetic structure. Usually we have one or two base changes, and they're typically base swaps that differentiate them, allowing for a couple of different protein sequence segments to be created. Okay, leg and arm length, hair color, and texture. So in humans, this gene has two different alleles or versions. So, and these are based on tandem repeats. So tandem repeats are just sequences of a gene that are exactly the same that just repeat over and over and over and over again in subsequent sequence. So you can actually see it in the gene. So there may be a gene that says GATTCA, GATTCA, GATTCA. That gene would have 3 tandem repeats. So, okay. And there may be another version of this gene that says GATTCA, GATTCA, GATTAC. And those two genes would be different alleles of one another, but they would both consist of having tandem repeats, okay? So again, it's just a short sequence that repeats itself within a gene. For the PER3 gene, the versions are related to the number of tandem repeats included in the gene. So I want you guys to think about that for a minute. If we have a gene segment where tandem repeats come into play and that differentiates the different allelic versions, okay? One will have a greater number of alleles or a greater number of tandem repeats and the other will not. That means there will be distinct variation in size for these two different genes, okay? That means that if we did something like gel electrophoresis with them, one of these repeats would move further than the other. So the larger segments would stay further behind on a gel plate, while the shorter segments would move further in the gel or more quickly because they have less weight to carry through the gel. We'll talk a little bit more about that. So the number of tandem repeats included within the gene changes the total length of the gene itself. So versions of this gene are physically different from one another. Again, they have different lengths and they function differently. So, and just so you know, the number of tandem repeats in a gene is an important factor for the regulation of gene expression. Okay. So here we can see a gene that has 5 repeats and then one that has 4. The one that has 4 has been associated with evening activity. The one that has 5 has been associated with morning activities. And again, differentiation in this number of tandem repeats ties in heavily with the function or the gene expression regulation. So, would there be any differences in expression if there were more than 5 or less than 5 repeats? Not likely, right? This would result in abnormality in sleep patterns. Could you have one of each version? Yes, you could. You could have the PERF5 gene or 5 tandem repeats and 4 tandem repeats on differential genes. So, what chromosome is PER3 located on? Chromosome 1. Is there a way to cheat the rate of the circadian rhythm? Yes, people do it all the time. There are people who have this evening gene, but they function more during the morning time than they do at night. So if their natural inclination is to sleep in and they're waking up at 7 o'clock, they'll have that 4 tandem repeat version of PER3. Okay? So here is the basic process for a historical isolation of DNA. It works first through the process of disruption, lysis, contaminant removal, and DNA recovery. So I'm going to kind of zoom in here for a minute if I can. So we can kind of see here, we have our genes that are added in, and we break the cell, okay? They're being kind of pulverized here. And then we extract the DNA, we add in a buffer solution, okay? We add in chloroform, and then we centrifuge, and then we have an upper aqueous layer that is removed. This upper aqueous layer contains contaminants. This step may be repeated multiple times, and then it leaves us with a solid sample of DNA at the bottom, okay? Then we would resuspend that DNA. So we add in 70% ethanol, we would dry that pellet out, and then we would suspend it in a different buffer solution. So this is the basic steps that make up the original PCR process. What we are going to be talking about in class is actually going to be a much more sped up version of this. So again, cell disruption, right? Cell disruption, lysis, contaminant removal, and DNA recovery. And again, contaminant removal is actually a process that is repetitive. So, and it's based on the size of what the contaminants are and the solution that they would be poured into, as well as what you would kind of use to remove those contaminants. I've seen it done a couple of different ways, but it is always a multi-step process. With advancements in molecular techniques, scientists have found ways to streamline this extraction process into a single step, where we would rub our mouths in class with a flathead toothpick, dip the cells in an extract buffer, and then boil to lyse the membranes and release the DNA via mini-PCR. Now, within mini-PCR, I do want you to realize we are still going through these steps. The machine is just doing it for you. The machine is doing these steps for you, so that you do not have to do these steps. It's been streamlined, okay? These steps are still happening. It's just the machine. So, again, basically with a polymerase chain reaction, we isolate DNA and we can make multiple copies of it, sometimes even billions of copies or millions of copies of DNA. This was invented by Carey Mullis. He was actually born in Lenore. And the outcome is a solution that contains amplified DNA. There are a couple of different components that need to be added into a tube in order to complete PCR. These are some of the reagents that you'll be using for your experiment. You'll have your DNA sample, in our case a cheek swab, primers. Primers basically show you where the attachment would occur for the extension of the DNA to happen. Okay, you'll have nucleotides that are added in, which also enable that extension to occur because they attach after the primers and allow for base pairing so that we end up with 2 subsequent strands of DNA that are complementary. And then we have Taq polymerase. Taq polymerase only functions at a very specific temperature. So it would have to function at one specific temperature or else it would be what? Denatured. So, and it is going to be used in these final steps here and a mixed buffer in a PCR tube. So basically we would start with one strand. It'd be denatured and ripped in half. That DNA strand would be annealed. We would have those primers come in and be attached. Taq polymerase would help come in and add in these base pairing sequences. And then the DNA would be extended out into where we have 2 strands. This would happen over and over and over and over again on an extremely rapid cycle. And we would end up with millions of copies of DNA in less than an hour. So again, we have the instructor DNA, pair of DNA primers, our forward and reverse sequences. And I want you to remember that these are read in opposite ways. So this would be the reverse sequence, right? This would be the forward sequence. And it would be because those strands have been peeled apart or denatured, and then they're being annealed, okay? So DNA polymerase, okay, in our case, again, Taq polymerase, DNTPs, all right? So again, those nucleotides are being added in a buffer to maintain and provide magnesium, which is necessary in the process of, doing this. So, or DNTPs, yeah. Yeah, or primers, sorry about that. And then free base pairs that you can kind of see happening here. See up in here. So, again, we'd have that one template DNA strand and rip in half. Each strand would become its own strand, and then we'd end up with 4 copies of the DNA or 2 copies of the DNA. These strands would be ripped in half, and they'd each make their own 2 strands. Those strands would be ripped in half. They'd each make their own 2 strands. So by the time we would get to our 4th cycle with one DNA sequence, we'd have 32 copies of that DNA. So how do we specify what part of DNA we want to amplify? So again, we would use those primers because they would only attach at certain parts, right? And that would happen during the annealing process. So again, denaturation, annealing, and extension. And this is kind of a visualization of what happens. These things can only occur at certain temperatures because these proteins can only function at certain temperatures. Okay? And if you extend beyond those temperatures or you go too low beyond those temperatures, it would slow the process down to where there was no point in doing this. So it's important to remember this is a very, very well thought out plan that Carrie Mullis worked through. And for the lab that we're doing this with, this would repeat in 30 cycles. So now, what applications are there in using purified DNA or amplified and purified DNA? Scientific investigations and understanding are really important, especially whenever we're working with a very limited sample of DNA and we don't have a lot of it that we can use. So we only have an extremely limited sample and we need multiple samples in order to run multiple experiments or see if there's variations in the DNA in multiple different ways. We would need a larger sample size. That's the biggest thing that this is useful for. Medical diagnostics with genetic diseases or predispositions, forensic purposes, and again, especially useful whenever you only have a set amount of DNA that has not been destroyed to use for a process. So whenever they do DNA testing for forensic classes or in forensic studies, they actually have to do PCR first a lot of times because they're starting with such a small sample. So we also need it for modification of DNA, right? For those experiments, again, we need larger sample sizes of DNA than what are being provided, and we don't want to hurt study subjects or things to extract that DNA. So GMOs, agricultural and livestock to generate consistent flavor, looks, and rates of growth. Right? Generation of transgenic animals for research and gene therapy, CRISPR, which is very interesting. So if you haven't checked that out, you should. So visualization of DNA with gel electrophoresis. How many of you guys have heard about gel electrophoresis or know what that is? How many of you guys have an idea of what gel electrophoresis is?
I do. I've done it before in class during high school.
Okay. Who's that? Sam. Yeah, so
Yeah. You're lagging out again quite a bit.
careful.
And she's gone again.
Whoop. All right. Sorry, guys. Can you see now?
Yeah.
Okay. Yeah, but basically, you remember whenever I had said, in class that you would be pipetting DNA, PCR DNA into these gel wells, and that it would be really easy to poke through the bottom of the gel, and you had to be careful. That's basically how you do it. So the first thing you would do is make the gel, okay, with different wells so that you can add your DNA in. After you made the gel with these different wells, you would add your buffer in. Then after you added your buffer, you would add in your DNA through the buffer. And this gel, it kind of makes a lattice that the DNA has to move through. So if you have longer pieces of DNA, they're not going to move as far. But if you have shorter pieces of DNA, they will move further through that lattice. So again, the gel is submerged in the buffer that add ions for the electrical current to pass through. Samples are then loaded into the wells with a ladder with known size values. And the ladder would be basically the control for the experiment, right? So it's a basis of comparison so that we can see where all of our size values are going to sit at. So we would add that in, and it would show us where on that gel a 50 band repeat or a 30 band or a 30 nucleotide repeat would be found. And you would be able to see it on the gel where they're sitting at. So, and again, that electrical current would be applied through the gel, and it would cause DNA, because it's negatively charged because of those phosphate groups, to move through the gel towards the end of the gel. Then the gel would be stained to visualize DNA. And then based on the latter, you could determine the size. And again, those shorter segments wouldn't move as far, or those longer segments wouldn't move as far through that gel. If I cut out again, just let me know and I'll pause for a second. And you can kind of see that here. We've got this lattice that's made up of this gel or agarose solution. And then we've got these DNA segments. So you can see that the shorter segments are going to move further through that gel than the longer ones. And what that would mean for the PER3 gene is those 5 tandem repeats would sit higher up on the gel, and those 4 tandem repeats would sit lower down. So it's very interesting. You may have 1,000 nucleotide base pairs at the top, but at the bottom you may only have a 20 nucleotide sequence for those segments of DNA. And you would it's not intrinsic, so you wouldn't think that you would look at a gel and you would see the greatest values at the top than the lowest ones at the bottom, but that's just how you read it. And again, it's because it's negatively charged, and we have that negatively charged DNA moving away from the negatively charged tip of the gel plate to the positively charged end. Okay? So, again, we'd be amplifying the PER3 gene to find out what genotype we have. So that allele with the 4 tandem repeats or the one with the 5 tandem repeats. So this week we're kind of going to be focusing on the processes of PCR and understanding that. The next week we'd be talking about gel electrophoresis and understanding the processes behind that. And then visualization of gel electrophoresis. So again, we have those different lengths of alleles. We run copies of our amplified genes on that gel to separate the DNA fragments by size, and would use a ladder, which is a basis of comparison, which has known fragment sizes, to estimate the size of the fragments we see and then determine if the DNA included has only 4 tandem repeats or 5, or is a mixture of both, which can happen in one person. So, week 2, we'd kind of be working here on this, and then this week we'd be talking about DNA extraction and running mini PCR, which goes through that process of denaturation, annealing, and extension. And then we have the gel visualization. Before we move on, and I have everyone kind of watch the procedure video, are there any questions at all about that? Is any of this confusing to anyone? I remember when I first learned PCR, it was really hard for me to kind of wrap my head around the fact that DNA strands are being split apart. And it's happening through a process where we have things like Taq polymerase that are so pivotal to the function of PCR. So if there are any issues with that, just remember when you're working through your DLNs, especially in the introduction section, you can always reference back to that PowerPoint and then take the portions of that PowerPoint that are bullet points and put them in sentence format so that you can explain and work through those first parts of the DLN. And just writing it out is going to help you remember it. So now we're going to kind of go here. I want everyone to watch the protocol video for exercise 6, cracking open the genetic code. Okay. I'm going to pause this. I should actually be able to not only screen share, but share my computer audio so that you guys can watch it with me in real time. So let's see. Now, would you guys rather watch this on your own or see if I can figure out how to share the computer audio with you? So
Can you do it?
I should be able to. Let's see. So, I was able to do it before. I might not be able to figure it out today. So what we might do is I might go ahead and have you guys click on that. But I want to add you learn it
You're breaking up again.
should be exercise 6, cracking up.
Excuse me. We cannot hear you.
Yeah. So I'm just going to give you guys about 18 minutes to watch that protocol video. Okay.
We didn't get the last like 45 seconds of that. You cut off.
It's okay. It's all right. All I said was to go ahead and click on As You Learn. And I'm going to give you guys about 18 minutes to watch the protocol video. Because my connection is coming in and out a little bit, I don't want to play it on my computer because I want to make sure you guys can all see it. Okay. So I'll just set a timer here in about 18 minutes. I'll go ahead and do that. And Mercedes, I see your text message, okay? So you just go ahead and do that real quick, okay? right. Everyone should be feeling be done with
Yeah, you cut off again. We can't hear you.
sorry about that, guys. But I'm gonna put you guys in breakout rooms here at about 7:20. I want you guys to leave the breakout rooms and come back to this okay? Now we're just kind of giving the breakout rooms a chance to close down here in about 30 seconds. I went ahead and attached my survey results into the chat for those of you who need that. The score was a 32, so definitely an evening person. And then we'll talk a little bit about the DLN, okay?
Hey, well, are we supposed to do the surveys? Like, do we have to do it?
Yeah, you do your survey, and then it should also ask for results from mine. I'm not sure if with the transition online, it's necessarily going to ask for yours or mine, but I think you need both, okay? So go ahead and just take it yourself, and then also include the survey results for me if the DLN asks for it. That way you have it and you don't miss any questions, okay? So how are you guys feeling about the DLN now that you've had a chance to look at it? It's pretty short. It's not a very long DLN. This is probably one of the shortest ones you'll have all semester. That being said, the one that you're going to do next week is going to be a bit longer. It's going to be a tidge longer. And again, next week, it's just going to be gel results for the most part. So you're going to be talking through the methods of the gel. You're going to be talking through, what that's going to look like as far as the results go and interpreting that. For this week, if I'm not mistaken, you really don't have a whole lot of results to interpret. And if I'm not mistaken, next week you shouldn't have to make a chart or a figure. It should just be more you talking about the gels and you turning the gel images themselves into figures. So that should be pretty straightforward for everyone. So I'm going to go ahead and share my screen real quick, and we're going to talk through the DLN. Hopefully this is still, recording. Okay, good. Now, let's kind of just click here. I did want to show you guys my scores, so if you didn't have it, here's that survey result. It's a 32. In between 16 and 41 indicates an evening type. So let's scroll here. And let's just take a moment to talk about this DLN real quick. It's very short, very brief, very to the point. And you shouldn't find it too difficult. So, again, consider what you know about yourself. This is more of an opinion-based question. Let's start at 1. So, you guys can all see my screen. So, again,
We can't hear you.
the cheek cell, they're easy to click. You guys should be able to hear it now. I'm going to go ahead and go back to that screen share and kind of show you that DLN and talk about that with you guys. So if you can't hear me, if it breaks out, just let me know so I can pause for a second for the connection to come back, okay? So buccal cell is a cheek cell. They're easy to collect because they shed often. And what kind of things are suspended in saliva? Well, for one, you may have food suspended in saliva and other contaminants. But, enzymes and proteins are also suspended in saliva. 3 components. You have that phosphate group, that nucleotide, and that ribose sugar. And the bonds that hold these components together, if I'm not mistaken, it's in the PowerPoint slide. So let's check that out real quick. But yeah, a lot of these first set of questions are in this PowerPoint slide. So you can kind of find those. And I'll just point them out to you guys so that you can work through things. But yeah, so here you go. Hydrogen bonding occurs between these monomers. So what are the 4 basic steps of DNA isolation and the extraction procedure? So again, you would go back to this PowerPoint slide here. You can kind of scroll down and you can see that boop, boop. Here, disruption, lysis, contaminant removal, and then DNA recovery. And then you describe how each step is accomplished. Again, make sure you're answering both portions of the DLN and the questions associated with it. What components are added into a PCR reaction tube? In your own words, describe the purpose. So you would want to do both parts of that. And again, you can kind of see that here, everything, and you describe the purpose of everything that's added into the tube. Okay, list and describe the 3 steps of PCR. Again, those steps can be seen here. Yep, denaturation, annealing, and extension, and you want to describe what those steps consist of doing. Okay. So describe the genotypes and associated phenotypes. Hint, there are 3 of each. So the circadian clock gene associated with sleep behavior preferences in human. And again, you've got evening, nighttime, and then mixed phenotypes. And you want to talk about the differences with 4 tandem and 5 tandem repeats. And then how some people lay somewhere in between. You can find a paper about PER3. So PER3 is actually a very interesting gene. It's been associated with mental health as well. There's actually one paper that's been attached in the As You Learn already that talks about anxiety. So for those of you guys with anxiety, they've done a lot of research about circadian clock genes as well as some developmental genes, and there's a lot of interesting information there for you. So you can kind of build off of that if you choose to. That's a great way to find a paper. And again, do you have a preference for morning activity, night activity, or both? And then that's just a, open-ended question. Input your MEC score. So that would be from this survey. Okay. And you'd want to take that paper and just paste it in. Indicate whether you're a morning type, intermediate type, or evening type and talk about it. And talk about if that's a preference you were expecting. Would you expect that you have 2 copies or 4 copies? And again, that's going to depend on whether you're evening or morning type, and you'll want to explain why. How accurately do you think the survey will be at predicting whether a study subject is genetically predisposed to be a morning or evening person? Do you think the survey is thorough? So again, you're just evaluating that survey. What types of evidence do you think help verify the association between tandem repeats and morning or evening being preliminary? So again, you could talk a little bit about how there are differences in transcription and translation that occur. And if you want to, you could find a paper about it to help with this question. And you could talk about how having those physical differences and the results of those differences associated with survey results could also help indicate that association. So, do you think your lab instructor's sleep phenotype is morning lark, night owl, or both? Therefore, what genotype do you expect them to have? I'll go ahead and tell you again, I scored a 32. And I've taken this before, so it is nighttime. And we'll be performing DNA extraction. What tissue are you still collecting for cells, and then how? It says buccal cells, and those are cheek cells, and it's coming from a swab. What reagents are you using to suspend the cells? What solution do you swirl the toothpick in? What is the goal of this reagent? Again, you can kind of follow that along in the procedure video and kind of see. Also, if you click through the PowerPoint, there is a specific part of the PowerPoint where it talks about the PCR reagents, as well as on As You Learn, there is a procedure protocol that'll kind of help you get an idea. So you can find that. So, and it should be a buffer solution. So you should be able to find that pretty easily. All right. Oh. All right. What is the purpose of placing the cell into a 95-degree Celsius water bath? Well, that depends on the step that you're at again, right? So are we extending that DNA out? So check again, see where that step is that requires that temperature, explain what the purpose of maintaining that temperature is, okay? And then talk about its association with that step along the process. So now, let's give it a second.
You're
Now it
breaking
says,
up
please
again.
describe in okay. Let's just give it a second. All right. Now, question 17. Please describe in paragraph format past tense and passive voice. And again, we talked about that and have those resources there for you online. How you and your peers collected the DNA sample and how you conducted the DNA isolation and extraction. You can either take the protocol that's provided to you on As You Learn, put it in sentence format, and exclude any preliminary information about results or what that may look like and talk about it, or you can write down word for word what you saw Parks do in the video without including any results that, or any idea of, or any discussion about results or what the DNA should look like, just what he was doing again. So list the reagents and amounts that are being added into your PCR tube. Again, you can see that in the video as Parks discusses it, or you can go in and you can find it in that protocol and As You Learn. What color is the solution in the PCR tube? What does this indicate and why is it important to check? So the color of the solution in the PCR tube is going to indicate the presence of DNA, right? And then you can talk about that a little bit more. And again, check that protocol sheet for the information there so that you can kind of put that in, and you should be able to find that pretty easy. So insert a picture below of the PCR tube. What you can do is you can just take a screenshot of Parks holding it in the video. So if you have a screenshot setting on your computer, you can go in, take a screenshot of the video where he's holding the PCR tube, or you can draw it, and I'll take both of those, and just insert it in and put a title below. That kind of just show, the PCR tube that's about to be inserted into the mini PCR machine. And then again, the picture of the mini PCR software and protocol that you're using to run the PCR below, there is this part of that protocol video where it shows the pre-denaturation, denaturation, annealing, and then extension, and then making the copies of the DNA that run and how long they run. And then you can kind of take a screenshot of that from the video and then paste it in here and then put a figure title below it. Okay. So you have the results of my survey, and you can kind of talk about that here. Right, and then there is a screenshot of the software. Oop, can you hear me still? Let's go back in here. You should be able to hear me still. Okay. But the PCR confirming that the cycle is successfully completed. At the end of the video, Parks does show that the cycle was completed, and you should be able to take a screenshot of the mini PCR software confirming that it's completed in that procedure video. And again, if you have a snippet tool in your computer, you can pause the YouTube video and take snippets of it and include it as a figure title. Or if you have a screenshot icon on your keyboard, you can do it that way. If you want to, you can also draw it out based on what you should see that it would look like on the video. So that'll be a good resource for you to check back to for a lot of these figures in this DLN this week, okay? If our PCR is run successful, what do you expect to have happened to the number of per 3 gene copies within the PCR tube? Well, they should have multiplied in the millions, right? So you're going to talk a little bit about that. If it was run unsuccessfully, what do you expect to have happen? You may have the same number of copies. The denaturation period could last too long. The DNA could be completely unusable. There could be contaminants. So whenever you run it on the gel, you'll be able to see contaminants next week. That's something that you're also going to want to talk about. And so contaminants in this case don't even necessarily have to be just contaminants as in proteins and that the PCR process wasn't run correctly. That's going to be the biggest issue that you guys are going to talk about when you talk about this question. But the other thing is like you have to make sure you're changing your micropipette tips. We're working with different solutions at different stages, right? So if you're not changing tips whenever you're working through those different parts, you may be adding in too little or too much of each part of those things required to run PCR in that tube. You maybe have not added enough DNA segments, so you could end up with a shortage in the amount of DNA that you need, or you could end up with something that's just going to look terrible in a gel and be really hard to interpret. So now, so what's the relationship between the two aspects of the researcher test and the study? We read the introduction section to determine what the purpose of this study was. And again, we are trying to amplify DNA and find out whether or not someone is a morning or nighttime person. So you can kind of talk about that a little bit based on the PowerPoint slide. So that should be a little bit helpful for you there. As you read through the sections,
You're breaking up again.
here we go. Let's make sure you guys can still see. Now, you should be able to. So here it's sort of the theory that having 2 copies of the 4 tandem repeat alleles is associated with evening preferences. So you should be able to find this from your pre-lab readings. So as you read through the results and discussion sections, right, of that. And you should also be, and he may talk about it some in the protocol video. But so, and again, the biggest thing that you're going to be able to do to associate that, again, it's just going to be survey results and also, you know, the PCR and evidence in conjunction with one another. But that should be something you should be able to find in your pre-lab readings. So according to the study, individuals with certain genotypes of the PER3 gene and or mutations are associated with sleep and or mood disorders. Using the data presented in the paper, discuss the possible links between the PER3 gene, sleep, and mood. And he's added one that talks about anxiety on As You Learn, and you can kind of use that to kind of talk about this here. So again, remember, you're going to make sure you initial. You'll lose 5 points if you don't. And do you guys have any questions, or did it cut out at any points where you didn't really get to hear me talk through something that you kind of wished you had been able to? At all? Well, I feel like this digital lab notebook is pretty straightforward. Again, most of the answers can be found on the PowerPoint slide. You can take the figures from the actual video using a screenshot tool on your computer and make figures out of them. And you should be able to do the same where it's asking you, like, for the beginning and ending of the PCR protocol or the sequences that you're using. Because you remember there's a screen that flashes on the YouTube video that kind of shows denaturation, annealing, and extension, and then all of that. Um,
Hey, you're breaking up.
So I feel like this is pretty straightforward. If you have any concerns, you can let me know. Again, just check that pre-lab reading again, and I think you should be pretty good. This is probably one of the shortest DLNs that you guys will have all semester. Again, the one you have next week is going to kind of make up for it. Just make sure you put really thorough answers down, and you maybe include like a paragraph per answer. That way, even if your answer is not completely accurate, I can still kind of understand what you're trying to say, because I know that going online is hard for a lot of you guys. So, if you guys want to go ahead and head out, you can. If you have any questions, you can stay and we can talk about it.
What do you consider like a paragraph? How many sentences?
I mean, just what you feel like you need to kind of cover the information. So if it's asking you to explain the associations between tandem repeats and then the PER3 gene, and you're looking through that pre-lab reading and you find a segment where it talks about it, if the segment in the paper is like 2 or 3 paragraphs long, you can condense it down to just the main points in a couple of sentences. If you feel like you need to use more than just a couple of sentences or a sentence or two to explain a concept or idea, feel free to. But at minimum, I would say just for every single response that you do, I would say probably about 2 sentences should be enough. at minimum, maybe 3 or 4 if it's a really more in-depth concept, like you're explaining denaturation, annihilation, and then extension. Because those can be a little bit harder to kind of grasp, and I do want to make sure when I'm looking through these, you guys do have a really good understanding of it. But just in general, this is probably a pretty short and sweet DLN, and you could probably get it done tonight if you wanted to. Probably in like the next 2.5 hours if you decide to like go really hardcore and include a lot of responses and a lot of answers to each question. But it's pretty short and sweet. So, yeah. Any other questions?
No, thank you.
You're welcome. I hope you guys have a wonderful day, and I hope that you guys have, that you're not too confused about anything.
Thank you.